And at the time, smallpox was killing hundreds of thousands of people across the globe. And Dr. Jenner took an experimental approach that really led to the development of the first vaccine. So it's a good thing, right? We had a proof of principle that we can make vaccines and cure human diseases through his actions. But what were his actions? Well, it was known at the time that milkmaids do develop cowpox, which was the cow version of smallpox. And when they developed that wasn't killing them. It was almost like providing them some kind of protection against smallpox. So what Jenner did is he decided to use the 8-year-old son of his gardener, James, and inoculated him with pus from the milkmaid that developed cowpox. So he developed some symptoms, but he wasn't really affected by the disease. After a while, Dr. Jenner took pus from a real smallpox patient and injected this little boy. And it turns out that he actually, through the initial cowpox inoculation, developed immunity to smallpox. So that's a great thing, right? Because he provided Jenner proof of principle that we can generate vaccines through this type of inoculation, and that led to eventually eradication of smallpox and the development of vaccines as a therapeutic approach for many human afflictions. But what's the problem with that? Would you do that today? I hope not. So then what to do, right? We can't really take little boys and inject them into experiments. So then how do we deal with this issue of trying to figure out what goes wrong in disease and how to come up with ways to develop therapeutic approaches for human diseases? And this is where animal models come in. So I'm sure we can all think of several experimental models, and I have a few examples here. This is probably the one that's, in terms of genetic makeup, the closest to us. That gene is about 97%, has 97% identical genomic DNA with us. But using this animal as a model is problematic in many ways, not only because of ethical concerns, but also think about the cost, the fact that they actually live quite a long life. So for example, if you want to study aging, that might be a problem, right? Because by the end of my career, the chimp might still be alive. So you want to choose your models carefully, depending on the problem that you want to study. And I would also say from the beginning that all models have advantages and disadvantages. And for example, this guy, rodents. This is the most common animal model in biomedical research. Mice and rats have taught us a lot about human biology and have helped us develop therapies. How about this guy? Do you know what this is? Baker's yeast, right? Makes awesome bread. Plants, worms, chicken, not the ones I chased, others, fish, and my favorite, the fruit fly. So one of the things that we might want to think about is, have we really learned anything from this? For example, my department head Joyce Schroeder talked to you about cancer last time. A lot of what we know about cancer cells, the way cells divide, have checkpoints, and those checkpoints go out of control, we learned from yeast. So all of these different models have taught us fundamental aspects of human biology as well as disease that I think remain underappreciated. And I don't have time to tell you about all the great discoveries that have been made with all these models, but I would like to tell you a little bit more about what fruit flies have done for us and what they can do for us even today. So when you look at this picture, this guy doesn't look very much like this guy, right? But they all come about from a single fertilized embryo. Yes, the fruit fly embryo is a little bit more oval than the human one. But if I told you that the way this single fertilized embryo comes to be an adult in flies, in humans, in cats, dogs, you name it. If I told you that it's actually the same cellular pathways and the same gene networks that take a single fertilized embryo to an adult. And what's most remarkable, and I want to share with you today, is the fact that the way the body plan develops from a single fertilized embryo and gives rise to the anterior or the head structure, to the posterior, the end structures, was really discovered in fruit flies, and this was recognized with a Nobel Prize in Medicine in 1995 to these three scientists, Christian Wolhard, who now has a lab in Germany, and Eric Wishaus, who is a professor at Princeton, as well as Ed Lewis, who passed away in 2004. These guys used chemical mutagens and generated large numbers of mutants in fruit flies and figured out how the body plan forms, how you go from a single fertilized embryo, a single cell, to all the cells in the body, and then how do you pattern the head all the way to the other structures of the body. And this is just a color code here to show you that when they looked at all these different mutants, and then they figured out how the individual mutations contribute to the development of the body plan, then they were able to put together a scheme, like a plan, if you want, of how different genes work together to generate an individual from the anterior all the way to the posterior. And this is one of my favorite slides that shows in a color-coded manner how you can generate a full adult, or in this case it's a developing mouse embryos. The same is true for humans. So the genes that generate the anterior part of the fly are the same that form the anterior part of the mouse or the human. The genes that generate the structures in the middle of the body are the same from flies to vertebrates, and the same is for posterior. So that was a pretty remarkable and unexpected finding that really demonstrated that this kind of developmental program and the utilization of gene networks is conserved from flies all the way to humans. These are just a few more examples that I'd like to share with you. Again, in schematic, showing that there are amazing similarities between flies and humans in terms of how we generate the body plan. Also, flies have a brain. Even if they have an exoskeleton and not a skeleton like ours, they do have the equivalent of a spinal cord. And they have neurons that project from the brain to the spinal cord, and then from the spinal cord to all the muscles in the body and allow them to move, just like our motor neurons allow us to move. Similarly, muscles and tendons are highly similar, and the gastrointestinal system is also very similar, and we've actually been able to learn a lot from studies of the fly gut epithelium, as well as the stem cells that reside in the gut, and have been beautiful drug screens have been performed using flies that are helping the development of therapeutic approaches for stomach cancer and gut cancer in humans. Another example that I want to share with you is just 2 years ago, the Nobel Prize in Medicine was awarded to these 3 fly geneticists that for their discoveries of the molecular mechanisms controlling the circadian rhythm. So the biological clock is important, right? It really controls our activities during the day, hopefully sleep at night. We all know that in today's society and as we age, this biological clock becomes a little altered, and there are sleep disturbances and sleep troubles that affect our health. What if I told you that the molecular basis of the biological clock was discovered by these 3 fly geneticists in flies? And this is just a simple schematic of one of the core components of the cycle. It's actually encoded by a gene nicely called period. I have to say that fly geneticists really like to give interesting names to genes, generally based on their biological function or the mutant phenotype. So in this particular case, the period gene actually oscillates with a biological clock. So this gene becomes to be made in the nucleus as an RNA, and and then it comes out into the cytoplasm and does its function of controlling the biological clock during the day. But then during the night, with the help of other proteins, makes its way back into the nucleus and regulates its own expression such that the next morning the cycle starts again. That's a pretty fundamental discovery, and it really, I think, shed important light on the relationship between molecular genetics, oscillation of actual gene expression, and our own biological clock. And there's some really interesting evidence that actually that the biological clock can even control the efficacy of chemotherapy drugs. So the time of the day, the activity of the biological clock, the oscillations of our gene expression profiles is actually tied in with physiological functions as well as response of the body in the conditions of various diseases. So, if I haven't convinced you by now that flies are cool and they have taught us some interesting aspects of basic human biology, I have a little summary slide here to show you why would anyone build a career on using flies to study human disease. And I'm used to the stares and I'm used to the laughter, so it's okay. I hope to leave you convinced that it's a worthy enterprise. So, first of all, 75% of human disease genes have fly homologs. The way I think of the fly compared to a human, it's like a little toolbox that you can buy at Walmart or Target, whatever you prefer, versus a real toolbox that you buy at Sears. So the fly gives you the ability to understand fundamental aspects of biology and disease, but in much, much simpler way, at a very reduced scale. So having this homology allows us to study the function of human disease genes in a much simpler model. I already told you that there are conserved genes and pathways that helped an adult develop from a single fertilized embryo. And there are some documented therapeutic examples. In the case of Fragile X syndrome, compounds that mitigated learning and memory phenotypes in flies were tested in patients and are in clinical trials as we speak. In addition, there have been at least 2 cancer drugs that are that have been FDA approved for skin cancer as well as men. This is a form of thyroid cancer that actually were initially discovered in fly models of disease and screened initially in flies. We also have a lot of fun in the lab. We have unparalleled genetic tools. Okay, imagine it's like being a kid in a candy store. You can take your favorite gene and express it in your favorite cell. You can knock it down. You can make an animal that has a mixed genetic background. You can make what I call GMOs, right? We can make humanized flies that express human genes. And that really allows us to use a very simple model to figure out the function of individual genes that could provide insights into human disease. They also have a short generation time. Graduate students love that because they know they can graduate in a timely manner, right? It takes 10 to 14 days to get the next generation, and it all starts with mom and dad mating. The female will lay fertilized eggs. These eggs, after 24 hours, will turn into larvae or maggots, if you want, and these undergo 3 different molting stages. They basically eat and grow, that's their job. And then after 3 or 4 days, morphogenesis starts, and the pupil will generate a new adult after 4 or so days. So in total, 10 to 14, plus and minus, depending on the temperature that they are raised at. They are also relatively inexpensive, at least compared to mouse models. And many scientists refer to them as being in the sweet spot for understanding human biology and disease compared to the cost and the investment that you make in this model. So, we've learned a lot by taking the approach of those fly geneticists that took that received the Nobel Prize, where they took flies, they fed them a chemical mutagen, and they did what we call a forward genetic screen, where you basically mutate genes at random, and then you observe what the consequences of that are, right? So you can basically work your way back and say, if I mutate gene X and I see this phenotype, that means that that gene X is important for the function that causes that defect, or phenotype as we call it. And some examples that I thought would be nice to show to this audience are mutations. So this is a normal fly eye. This is a fly that carries a mutation in a gene nicely called eyeless. So when you have a mutation in the gene eyeless, there is no eye. This fly is blind. The human homologue of this gene, also called eyeless or PAX6, when it's mutated, causes this disease called aniridia. Although there is a crystalline, there are no photoreceptors and there is no retina. So in this particular case, there is blindness caused by a mutation in the same gene, and these mutations were initially identified in the fly. Another example that I wanted to give you is mutations in a gene called hedgehog. I told you that fly geneticists like to give funny names. This comes from the fact that when you mutate this gene, the fly embryo is so spiny and hairy that it really looks like a hedgehog, hence the name. So when you have a specific mutation in hedgehog, you generate a duplication of the wing that looks like that. The same mutation in humans causes polydactyly, with the duplication of the thumb, as you can see here. So at least visually, the similarities are remarkable, and they go all the way down to the same gene and the same mutation in the two different species. One of my favorite uses of fly, in addition to generating mutants and looking at the consequences of this mutation, is using flies to study behavior. I would say mind and behavior are really fascinating, but also very difficult to figure out. And so one of the ways in which flies have been used for behavioral studies is to study aggression. And if I can make this work, I can start this movie maybe. Colin, would you be able to help me, because this is your laptop? The reason I chose this is not because it's one of my favorite behavioral assays, but also because in my own lab we are starting to introduce these aggression assays to study behaviors of fly models of dementia that we are developing. One of the presentations of patients with dementia is that they are aggressive and exhibit different social behaviors. And so, does it work? So, oh, but I will need the sound from the laptop. Will that work? So I would love to be able to adapt this kind of assay into my lab and study the effect of dementia-linked genes in humans all the way into the fly and see if that causes aggressive behavior. And we have some exciting preliminary data that shows that that is actually the case. You won't play because it's a PC? All right, that's unfortunate, but maybe what I can do is I can just provide it later, like a link for people to look at. It shows two males fighting for a female, and it shows the different stages of how they fight for that female. You know, they first get to know each other, they chase each other, they give each other a few punches, and then they end up, one of them is the winner and throws the other one off the podium, and I guess gets the female, right? So I'm sorry you can't see that. It was really cool, and it also had cool music, but you can imagine. All right, so this is what I've shown you so far, is how you can go from fly to human by generating mutants and looking at defects that those mutations generate and imagining behaviors that flies exhibit. But we can also go the other way around. And I think this is really important in today's research, and we are adapting this quite a lot in my lab as well, and many other labs around the country and around the world, where it's really important to understand what goes wrong at the genetic level or at the molecular level in a patient with a particular disease, say ALS or FTD, frontotemporal dementia, which is what we focus on in my lab. But let's say you find a change in the DNA. It's really hard to figure out if that change causes the disease or causes aspects of the disease. With the exception of mutations in the Huntington gene that causes Huntington's disease 100% of the time, there is really no other example where you have this perfect correlation between a mutation and the disease. Essentially, mutations are risks, confer risk of disease, because everything is so much more complicated. There are so many other changes in the DNA that contribute to the effect of that mutation. And in addition, there is the interaction with the environment that changes the risk that a particular mutation has. And one of my colleagues in the spring, Ryan Gutenkust, will come and talk to you about 23andMe and about how we can interpret all these changes that we can now identify at the genetic level. But essentially, I don't know how many of you have done 23andMe here. I did. Did you find that satisfying? Did you? It's somewhat satisfying. They told me I'm from Romania. But not from the beginning. After a few years, after that, I think they got more data. And they also told me I have like 0.3% Asian, so I'm trying to figure out which one of my parents went wherever, but anyway. But in terms of the actual changes at the DNA level, it's interesting, but for the most part, we don't really know what to do with that information. And we don't know exactly how to interpret those changes as cause of disease, for example. So here's where flies come in, or other models, but of course fly is my favorite. You can make an avatar. How would that work? You can basically take a change in the DNA that's in a human gene and make that exact change in the fly gene, or you can take that human gene with that exact change and put it in the fly. We call that a fly avatar because it sounds cool, but it really is a humanized fly. And we've actually done that. I've been working with a consortium that has been helping a patient who has ALS. He has a very slow progressive form of ALS. And none of the common mutations that cause ALS, he didn't have any of those. And so colleagues of mine from this consortium sequenced the genome of this individual, and they found a couple of changes compared to his parents, whose DNA was also available. And so that gave us some hints that these two are likely causative of his slow form of ALS. So I had colleagues who put these mutations in yeast and look at phenotypes. There wasn't much going on there. So then we took the mutation in one of these genes that we thought could be causative of his ALS, and we made a fly with that exact mutation. And guess what? That fly exhibited locomotor defects and didn't have such a severe effect on survival, which kind of nicely matched the fact that this individual had a very slow progressing form. Now, we don't know the whole story, and it's really difficult to make a fly with the exact all same changes in his DNA. But we took the one that we thought was most important, and that caused locomotor defects and synaptic defects in our flies. So that's as far as you can go to say that that mutation is likely causative of this individual's ALS. And then, as with other colleagues in this consortium, we were able to feed these fly avatars specific drugs And then some of my colleagues generated motor neurons from these individuals' skin through induced pluripotent stem cell technology, and we kind of worked in parallel and tested and identified compounds that were improving the fly locomotor defects as well as the motor neuron phenotypes in a dish derived directly from the patient. So this is kind of where this type of model system experimentation and disease modeling can help. Of course, not everybody can afford that, right? But eventually, this kind of modeling can become cost-effective and relatively rapidly can identify compounds and strategies that can mitigate disease. So next I want to tell you a little bit about my work that's going on in my lab. For the past 10 years, we've been focusing on studying the molecular mechanisms that underlie amyotrophic lateral sclerosis, or ALS. These are some examples of famous people. I cannot find a famous woman that died of ALS. It's true that it affects more males than females, but these are also, I can only find famous males, not famous females, so that's I don't know. Anyway, the disease is also known as Lou Gehrig's from this guy, and it's a progressive neurodegenerative disorder. By pharma standards, it's considered a rare disease, but when you meet the patients and their families, it's never a rare disease when it's close to home. Age, just like for all neurodegenerative disorders, is the biggest risk factor, and most people develop symptoms after 55 years of age. What's most devastating about this is that it's very rapid, and I don't know if any of you have experienced or have met a patient with ALS. It's very fast and it's very dramatic, and essentially the muscles melt away as you look at them. And their locomotor function and their ability to move basically goes down relatively quickly and very dramatically. Now you might wonder, how about this guy? For a while, it's been thought that maybe he didn't have ALS. In the end, it was thought that he did have ALS. And I hope that someone stole a hair or a piece of skin and is sequencing him, because it will be really cool to figure out what was in his genetic makeup or his genetic background that made his disease a slow progressing form. There are only 2 FDA-approved drugs for this disease in the United States. One was approved maybe 10 years ago, the other one was approved 2 years ago. At best, these drugs improve survival by 2 months. And in most cases, they have no effect. So we're at the point where there is a real need to make a concerted effort to try to figure out what's going wrong in this disease and how we can stop the degeneration. In terms of what happens, this disease is very heterogeneous. We have upper motor neurons that are located in the brain and project into the spinal cord. We also have lower motor neurons that are located in the spinal cord and project all the way to all the muscles that control everything from breathing to movement to speaking. This disease comes in 2 different forms. A familial form that affects about 10% of patients, and a sporadic form that affects 90% of patients. And we do know that there is a genetic basis. So this is a graph that shows you in time how new genes have been discovered. The very first one is called SOD1. This is not exactly updated, but it's almost close, almost updated. And you can see that we know of many genes that, when mutated, can cause ALS. But 90% of patients have either de novo mutations or an unknown cause. And what's really difficult is that it hits people out of nowhere, and it seems to affect more veterans and more athletes, perhaps because of injuries, but we're not sure. There are also environmental causes that we don't really understand. Smoking is listed, but no one really has a clue what is it about smoking. Yeah, sure, there are toxins in the cigarettes, but it's unclear what the reason is. And again, I think this is an opportunity to really try to understand what is it in our environment that can cause motor neuron disease or neuronal dysfunction. What is really remarkable about ALS is just like for a long time, it's been thought that it's just a motor neuron disease. But in the past 5 to 6 years, less than 10 years for sure, it's become clear that ALS is not just a motor neuron disease, and kind of like autism is part of a spectrum disorder, with ALS, motor neuron disease on one end, and dementia at the other end. And the data supporting that comes from genetic discoveries. So if you look at one end, an individual with mutations in SOD1 will only get motor neuron disease. An individual with mutations in tau will only get dementia. But individuals that may have mutations in any of these genes in between, they can get either ALS or FTD or both. And so this has become apparent by looking at families in which one member has developed ALS, another FTD, another both. And even more remarkable, when you look at post-mortem samples, pathologists take slices of spinal cords or brains of patients that passed away from ALS or FTD. They find generally this kind of clumping of protein or protein aggregates. And the protein that is present in these aggregates is called TDP-43. It's a protein that binds RNA, and that's how we became interested in it, because I'm interested in RNA biology. But we're not going to go into the details of that. I just want to point out that 97% of patients that die of ALS have these protein clumps that contain TDP-43. And 45% of patients that die of dementia have these protein clumps that contain TDP-43. So you can imagine that if you focus on understanding everything about this protein, what is TDP-43 doing in a normal neuron? What is it that goes wrong in disease that this protein, even if it's not mutated, okay, this is the weird thing, it's not mutated, but ends up in these aggregates. And these are those aggregates that in Alzheimer's people have tried to, disrupt for a long time without much success, I might say. And there is still controversy about whether these aggregates are protective or they're damaging, because that's really good evidence to support both. What's even more interesting is that in the past 2 to 3 years, it's become apparent that TDP-43, it's not just present in ALS and FTD, but also in Alzheimer's. Between 10 and 50% of Alzheimer's patients contain, yes, amyloid plaques, but also TDP-43 aggregates. Parkinson patients contain TDP-43 aggregates. More recently, football players that have suffered injuries and passed away from chronic traumatic encephalopathy have TDP-43 aggregates. Soldiers that may have suffered traumatic brain injury or TBI have TDP-43 pathology. So this has emerged as kind of a new paradigm in the field of neurodegeneration, where you find this one protein that somehow knows that there's stress around and the neuron is diseased and ends up in these aggregates. So again, if we could figure out what is TDP-43 doing and how it ends up in those aggregates, and what are those aggregates really doing to neurons? Are they bad? Are they good? Could it be that some are bad and some are good? If we could figure all that out, we could perhaps stop the process of neuronal dying. Okay, so I hope I convinced you that TDP-43 is important. Yeah? All right. So then how to study it in a fly? Yes, flies have TDP-43 homologue, but how to develop a good model? We decided, and I'm running out of time. We decided to make a fly model. I call it an avatar or a GMO, whatever you want to call it. I could make a shirt and go to Whole Foods and say, I make GMOs for a living. That's my dream. So how do you make a fly model that could be good to study ALS? We took this human TDP-43 and we put it in motor neurons, and just in motor neurons, because those are the cells that die in disease. And what you're looking at here, every little dot is a single motor neuron in a normal fly, and this is what happens when you express TDP-43 in motor neurons. You see there are fewer neurons because they're dying, just like they do in human patients. This is a picture of synapses. You can tell that they look different in the fly that expresses TDP-43 in motor neurons. Compared to a well-fed control fly in the lab that can live up to 100 days, when you express human TDP-43 wild type or mutant in motor neurons, you can see that they live less, just like patients live less. And will this work? Or do we not have the movie capability again? What I would show you here, if I could, and Colin is going to try, is that we can measure locomotor function in these animals that express human TDP-43. And on the left there is a larva that has ALS. Oh, it works. So take a look at that, and you see, even if you don't look at flies like I do every day, hope you can see that this guy is dragging its posterior end because it's paralyzed, okay? But, can you help me with the second one, please? If you look at the one on the right, this one has the exact same genetic makeup. So it's an ALS larva, but we fed it a drug that we think is protective. So I don't know if you can tell, but you see how it's moving a lot better? So it's actually visual, and undergraduate students love to do this, right? They put the larvae on a drug, food, and then they do this assay, and they actually quantify, and we can measure with numbers the amount or the effect that a particular drug has on this locomotor dysfunction. And we can do other assays, but I just wanted to show you this one as a visual example of how you can see or how you can generate an improvement in a locomotor defect and a paralysis in an ALS fly. So what's the deal with these neurons? Why are they so susceptible? Because patients that have familial ALS, they have the mutation throughout the body. So what's so special about motor neurons? And I'd like to pose you with a challenge that they have. The longest motor neuron in our body is located in the spinal cord and goes all the way to the toe. It's about 1 meter long on average. If you were to draw this to scale, the cell body would be the size of a basketball, but the axon will go 1.5 miles all the way, at the place where I got lost when I got here, okay? So it's a huge challenge because you have to do, you have to take proteins there, you have to take RNA, you have to keep everything in order. It's like having a well-organized factory. It's like Amazon, right? Like they have those robots that move everything. This is what a neuron needs to do to function correctly. 1.5 miles, that's a lot if you just make everything in this size, right? So indeed, what we find to be a problem in these neurons with a long axon are some traffic jams, proteins clumping, mitochondria clumping, not being able to make energy. Mitochondria are the factories of the cell. We also see some broken roads, especially towards the end. And if you have a traffic jam and then the road is broken, you can imagine that you can't take to that end place everything you need to make that neuron function correctly. And eventually, that neuron will suffer back and will start dying from this end towards the cell body. This is just a model for how clumping may occur, but I'm going to skip that. So next I have some real data to show you. We call ourselves fly neurosurgeons because you take that little larva and you can dissect it under the microscope and you open it up like that, and here's the brain, here's the fly spinal cord where all the neurons are located, and here are the axons coming and forming contacts onto the muscle that coordinate that movement I showed you in the movie. So we can look at the motor neuron cell bodies, we can look at their axons, and we can look at the neuromuscular connections onto the muscle using a confocal microscope. This is about 0.2 micron resolution. And all I want to show you here is that this protein, called FUJ, because the gene that encodes this protein, when you mutate it, causes a loss of axons. And in German, FUJ means gone axons. Okay, that's where this comes from. So if you look in a normal fly, here's the distribution of this protein. When you look in our ALS larva, you see a lot more protein here, and you see a lot less protein when you go down to the synapse. And this is quantified here, you don't need to worry about that. But one of the things this was our very first molecular target that we identified. And the question was, what does this mean? Is this really relevant to studying human disease? So how would you go about that? You discover it in the fly, you figure out what it does in the ALS fly, and then you ask, do we see a similar defect in human patients? So with collaborators from the Neurological Institute, we obtained spinal cords, and these are spinal cords from healthy controls, and this is a spinal cord from an ALS patient or an individual who passed away from ALS. And the staining in brown here is for the human homologue of the fly FUJ protein. And I hope you can see there's more here in the cell body compared to the control, just like in our fly. So this was just the first indication that we can use the fly genetic and molecular tools to predict what might be wrong in humans. There are over 30,000 genes in the human. We can't take them one at a time and figure out which one is dysregulated. But you can use a simple model and use this molecular and genetic power to figure out what are the most important genes that are altered in disease and then validate them in a human, hence the fly-to-human approach. So in the interest of time, I'm going to skip some slides, but I want to show you a little bit of the work we've just published a few months ago. ALS patients are thought to be hypermetabolic. For a while, I couldn't figure out what that means, and I thought it just means that their muscles are melting away. No matter how much they're eating, they're not gaining weight. And then there's some evidence that patients that have a higher fat content, or even patients with diabetes that have higher triglycerides, fare better when they get ALS. And then there was a small pilot clinical trial conducted at Mass General Hospital in Boston that showed that if you feed an ALS patient a high-calorie diet that has more carbs than other components, those patients do pretty good. But it was very small, and we couldn't draw any conclusions. So we decided to test this in the fly, and I just want to remind you all that our brain, although small compared to the rest of the body, it takes 20% of our glucose consumption. That's why when I write grants, I eat a lot of candy, because I need that power. That's my justification, at least. So for cells to make energy, they take in glucose, and they make energy, or ATP, in two major ways. One is through a process called glycolysis that breaks down glucose all the way to something called pyruvate, and it generates 2 molecules of ATP. A lot of work for 2 molecules of ATP. Then the result of glycolysis goes through the mitochondria, which is the real power factory of the cell, and takes that product and makes 28 molecules of ATP. The problem in neurodegeneration is that these mitochondria don't work very well. So then, what happens when we take those larvae and we send them off to a company that can look at different metabolites? They told us that the end product of glycolysis is upregulated. Okay? And then it was really interesting because a patient study showed that the same products of glycolysis are also upregulated. So again, all right, the fly showed us that we see similar things as happen in patients. So what should we do with that? For glucose to be processed, you need many enzymes, but there is one that's really special. It's called PFK, or phosphofructokinase. And this enzyme, it's what we call rate limiting. The more of it you have, the more glucose you can process. And so what we did is we took flight tissues, spinal cords from patients, and patient-derived motor neurons from skin cells. And I'm not going to bore you with the details of the experiment, but look above this bar. Except for this, everything is higher. That is consistent with the fact that at the molecular level, glycolysis is increased. Okay? It's higher in flies, in patient spinal cords, in patient motor neurons. But what does that mean? It could mean that neurons upregulate glycolysis, that this mutation, or TDP43, upregulates glycolysis, and that causes neurons to die. But it could also mean that neurons upregulate glycolysis as a compensatory mechanism to survive. So which one is it? We scratched our heads, we talked a lot, and then we said, let's do the experiment. So how should we figure this out? So in flies, we can do a couple of things. We can feed them different diets. If you feed a fly a high sugar diet, do you remember the larval turning experiment? The more paralyzed they are, the longer it takes for them to move. If you feed a normal fly a high sugar diet, it becomes sluggish and eventually gets diabetes. If you feed an ALS larva a high sugar diet, you see how this turning time goes down and their lifespan increases. Look at light pink to dark pink and light blue to dark blue. So giving the ALS animals more sugar was good for them. But this is a brute force approach, right? Sugar could get into any cell and could improve movement because it affects muscles or something else. So then here's where genetics comes in. I told you we can manipulate genes in a cell type specific manner. So we took the ALS fly and we put human glucose transporter just in motor neurons. And what that does is it allows those diseased motor neurons to take in more sugar. And here's what we got. The same effect. The larval turning time went down and the lifespan got increased. So if we simply make motor neurons that are diseased take in more glucose, either by giving them more glucose or genetically putting in a glucose transporter that sucks in all the glucose that's available, we can improve the outcome while we make genetic controls worse. So I'm going to try to wrap it up. Yeah? So wouldn't it be wonderful if we could just figure out what goes wrong energetically in a neuron and then try to rebalance that? So we know that mitochondria don't work very well. So the cell is starved because you don't have that high capacity to generate ATP or energy. So what does the neuron do? Oh, not all my animations worked. I had a spoonful of sugar. Sorry. What neurons do is they upregulate the capacity to take in sugar. So if you basically provide them with a spoonful of sugar, you can improve their ability to generate energy, you improve neuronal function, and we've done a lot of experiments to show that the actual synaptic function is improved. So it could be as simple as that. But one of the things that I want to leave you with, there's a couple of things that I'd like you to leave this place with. One is that flies are cool. Hopefully you agree with that. Second is that perhaps there are more similarities between the different types of neurodegenerative disorders than we previously thought. On one hand, we have this TDP43 pathology that's common to all these different diseases. You also have, if you basically look at a diseased neuron, there's mitochondrial dysfunction, there are protein clumps, and there are those breakages in the road. You see that in Alzheimer's, you see that in ALS, you see that in dementia, you see that in Parkinson. Even if you start out with a different mutation, you basically end up causing the same deficit. So perhaps we need to change the way we view this, and rather than focus on a single gene and single disease, we can start taking more big picture view and more big picture approaches that can help us understand what happens in all these disorders, what is in common, what might be different, and how can we intervene to stop the disease process. The other thing that I'd like to leave you with is that although flies are not humans, and I call them flies are little people, that's what I tell my students, but, of course they're not, right? But perhaps we can go back to this guy's poem. Am not I a fly like thee, or art not thou a man like me? And with that, thank you for your attention. And I have some flies here that I can show you what they do when we're done. And lastly, the work is done by these guys. These are my students who have done all the work that I showed you, and also the funding agencies that have funded this work. So many thanks to them. Thank you for your attention, and I'm happy to take questions.
Does anybody have any questions for Daniela? Bring the microphone to you. Do you have that?
Have you tried to replicate any of the environmental implications in your lab on the flies?
We're trying. I would love to. The mutation I told you about, the fly avatar that we make that harbors the mutation of this patient with ALS, affects copper homeostasis. So somehow copper is important to motor neuron function, and this particular mutation affects the way motor neurons handle copper. So I would love to actually study this, and I was thinking we are in a copper state. It would make a lot of sense to actually look at that, to look at the effect of environmental factors like copper, but nothing beyond that. It's just in the wish list.
Are there applications that people thought about in all of this for using CRISPR technology?
We are using CRISPR. Do you mean, can we use CRISPR to generate modified flies? Or, yeah, we have generated CRISPR. It's very elegant, but I would say it's not as easy as the news tell you. When people tell you you can do it in your garage, no, you can't. Okay? It's actually really hard. But we can use CRISPR, and we actually have such a model where human TDP43 has been CRISPR'd in to replace the fly TDP43. So then we just have the human gene to look at what that causes in terms of motor neuron phenotypes and other neuronal phenotypes. Yeah.
Back here.
You mentioned that there were 2 drugs that were available for ALS patients. Were these drugs used as a cure or just to make the patient, make it easier for them to die?
There is no cure. It doesn't, they don't make the patient, it doesn't make it easier to die either. It just, in principle, is supposed to mitigate or slow down the disease process, but it doesn't really do that, except for in a few patients there is maybe 2 to 3 months increase in survival. So one of the drugs is affecting signaling from neurons to muscles, and the other drug deals with oxidative stress, which is increased in aging and other disease neurons. But there's no cure. And in the end, even if a cure would be difficult to find, there are definitely ways that we can think of that could stop the progression. And I think that's a very reasonable and realistic goal.
I was just curious if ALS is known to be prevalent in any other species.
Dogs get ALS. There is also a spontaneous mouse mutation called the wobbler mutation. It affects calcium signaling in neurons. But yeah, dogs get the form of ALS.
I have a two-part question. First part is, are you suggesting that a high sugar diet is helpful?
High sugar diet is what?
Helpful?
Yes, I am. And I'm actually working with clinicians really hard, trying to convince them to do a trial. And I'm working really hard on that. So when I first told them, they looked at me a little funny. And then I talked to them about this pilot trial. And they said, okay. Then I showed them the flight data, and they said, okay, but we're not convinced. So I said, what would convince you? And they said, we want to see the same molecular changes in patient tissues, which now we have. So the challenge is that it costs money to do a larger trial, and most agencies or companies that fund trials prefer a drug to a diet, because as we all know, nutritional interventions, you hear one day that this thing is good, the next day it's not so good. So there is a little bit of a bias in the challenge to fund a nutritional study, but it will happen. I'm working on it.
Second part of the question is, you say that 20% of the energy in the body is used by the brain. And so if you're short of I'm trying to figure out how to exactly express this question, but if you take extra sugar or don't, that's going to affect the brain function, I guess.
So it's all a matter of balance, and that's where the hard part comes in. If an individual is healthy, too much sugar, just like in a healthy fly, it doesn't do good. Too much sugar in a healthy environment causes oxidative stress and damage.